You have probably experienced it yourself. A meal you ate once, years ago, that made you ill afterward. Perhaps it was a particular dish at a particular restaurant. Perhaps it was something you cooked at home, or ate at a party, or were given as a child. You recovered quickly. The food itself was almost certainly not responsible. It was more likely a coincidental virus. And yet, the thought of that food, even now, produces a faint but unmistakable aversion. The smell of it, the texture. Something about the taste is stored in your body as a warning, and the warning persists years after any rational basis for it has passed. This is taste aversion, and it is one of the most striking demonstrations in all of psychology of the fact that biology shapes what can be learned, how quickly it can be learned, and how long it will last. For most of the mid-20th century, the dominant assumption in learning theory was that conditioning was essentially general purpose. Any neutral stimulus could be associated with any unconditioned stimulus, given enough pairings and the right conditions. Pavlov had shown that a bell could be conditioned to produce salivation. Watson had shown that a rat could be conditioned to produce fear. There was no principled reason within the standard framework why any stimulus should be more or less conditionable than any other. John Garcia, a researcher at the Naval Radiation Laboratory in the 1950s, was about to find a very principled reason. Welcome back to the Brightest Experiments. Today we are inside one of the most important boundary-pushing discoveries in learning science, a finding that was initially rejected by the leading journals in the field, dismissed as impossible by the most prominent researchers of the era, and eventually recognized as one of the landmark results of 20th century psychology. Garcia was studying the effects of radiation on rats, specifically on their eating behavior. Rats that had been exposed to radiation became ill, and Garcia noticed that they subsequently avoided the food they had eaten before the radiation exposure. The association between the taste and the illness had been learned, even though the illness had not occurred until hours after the food was consumed. This observation stopped Garcia in his tracks. Standard conditioning theory held that the interval between the conditioned stimulus and the unconditioned stimulus needed to be very short, a matter of seconds, for conditioning to occur. Associations between events separated by hours should not form, and yet here they were, reliably and powerfully. Garcia designed a series of experiments to investigate this systematically. The basic design was simple. Rats were given flavored water to drink, then made ill, either through radiation exposure or by injection with a substance that produced nausea, either immediately or after a delay of up to several hours. The question was whether the rat would subsequently avoid the flavored water. The results were unambiguous. Rats developed strong aversions to flavors associated with illness, even when the illness had been delayed by several hours. A single pairing was sufficient. The rat did not need multiple trials, as standard conditioning procedures required. One experience of tasting something and becoming ill, even hours apart, was enough to produce an aversion that persisted indefinitely. Garcia then tested the boundary conditions of this effect with a series of elegant experiments. He compared learning across different combinations of stimulus and consequence, taste paired with illness, taste paired with electric shock, audio-visual cues paired with illness, audio-visual cues paired with electric shock. What he found was a striking selectivity. Rats learned taste-illness associations with great ease and across long delays. They learned audio-visual shock associations equally well, but taste-shock associations and audio-visual illness associations were extremely difficult to establish, even with short delays and many pairings. The pattern was not random, it was biologically meaningful. Tastes are the signals most relevant to food quality. If a food makes you ill, the taste is the most informative predictor of that outcome. Audio-visual cues are the signals most relevant to external threats. If something in the environment hurts you, what you saw and heard is the most informative predictor of that threat. The rat's nervous system was not a general-purpose learning machine that could associate any stimulus with any consequence. It was a biological system shaped by evolution to learn specific associations more easily than others, the ones most likely to have been ecologically relevant to its ancestors. Garcia called this phenomenon belongingness, the idea that certain stimuli and certain consequences belong together, in the sense that they are prepared to be associated. The concept was not entirely new. Martin Seligman, working independently, had developed the theory of preparedness to describe the same phenomenon in a broader context. But Garcia's experimental demonstration was the clearest and most detailed available. When Garcia submitted his findings to the major journals in experimental psychology, they were rejected. Reviewers described the results as impossible, as inconsistent with established learning theory, and therefore almost certainly the product of methodological error. The long delay intervals were implausible. The selectivity of the effect was theoretically inconvenient. One reviewer reportedly said that to believe Garcia's results would be like finding birdshot in a bottle of Bordeaux, physically impossible and therefore not worth investigating. Garcia persisted. He replicated, refined, and extended his findings across the following years, and eventually the weight of evidence became impossible to dismiss. By the 1970s, taste aversion conditioning had been accepted as a genuine phenomenon, and its implications for learning theory were being worked through with the seriousness they deserved. The scientific consequences were significant. Garcia's findings forced learning theorists to abandon the assumption of equipotentiality, the idea that all stimulus-response combinations are equally conditionable. They established that biology places constraints on learning, that evolution has pre-equipped organisms to make certain associations more easily than others, and that the efficiency of learning depends on the fit between the type of association being learned and the ecological history of the species doing the learning. The practical applications of taste aversion conditioning are equally significant. Wildlife managers have used it to protect livestock from predators, treating carcasses with a nausea-inducing substance and leaving them where coyotes or wolves will find them, so that the predators develop an aversion to livestock prey without being killed. The technique works. It changes behavior through learning rather than through elimination. In medicine, taste aversion has been recognized as a significant side effect of chemotherapy. Patients who eat before a treatment session often develop strong aversions to the foods consumed because the nausea produced by the treatment is associated with the taste. Knowing this, oncologists now recommend that patients avoid their favorite foods before chemotherapy, and in some cases give patients a novel, dispensable food to consume beforehand so that the aversion attaches to that rather than to nutritionally important staples. For understanding your own psychology, taste aversion is a vivid demonstration of a broader principle, that not all learning is equal. You are not a blank slate on which any association can be written with equal ease. Your nervous system came pre-configured with certain biases, certain types of experience you are prepared to learn from quickly, certain associations you will form from a single exposure, certain responses that will persist long after the original event has been forgotten. This is not a limitation. It is the record of what your ancestors needed to learn quickly and permanently in order to survive. John Garcia set out to study the effects of radiation on rats and discovered instead a fundamental property of learning itself. His work was rejected by the field, confirmed by replication, and eventually recognized as essential. He received the Distinguished Scientific Contribution Award from the American Psychological Association in 1979, formal acknowledgement that the finding the journals had dismissed as impossible was, in fact, one of the most important in 20th century learning science. The taste you never forgot is not irrational. It is your nervous system doing precisely what it was designed to do. Next time, we move from the learning of aversions to the learning of language, one of the most remarkable achievements in human development. Roger Brown and Ursula Bellugi spent years recording the spontaneous speech of young children and discovered that language acquisition follows universal patterns, that children are not simply imitating adults, but constructing grammar from first principles, and that the developmental sequence of this construction reveals something fundamental about how the human mind works. That is coming right up. Leave a comment. Have you experienced a taste aversion? And does knowing its mechanism change your relationship to it at all? I'd love to hear. Subscribe so you're here for the next episode. I'll see you then.